Hey guys, Mr. P. In this video we're going to talk about cell cycle regulation. So we've talked through all things cell cycle. We've talked through the phases of the cell cycle, G1, S, G2, and M phase. We've talked about all aspects of the subphases of mitosis. We've talked about the differences between mitosis and meiosis. We've discussed things like genetic continuity and mitotic division. We've also talked about meiotic division and how meiosis uses crossing over and independent assortment to increase genetic diversity. And so now we're going to wrap up this entire playlist by talking through cell cycle regulation, which is how the cell cycle is regulated. We're going to focus on three objectives. The first objective is D2.1.15, which is control of the cell cycle using cyclins. So we're going to talk specifically about cyclins and how those cyclins aid the cell cycle or regulate the cell cycle by producing checkpoints. D2.1.16 is consequences of mutations in genes that control the cell cycle. So there are gene regions, gene coding regions that are going to help control the cell cycle, and just like any other gene, they can become mutated or can experience mutations. And so we're going to talk about the consequences of those mutations and how that ultimately fails to control or overregulates or underregulates the cell cycle. And the last objective is D2.1.17, which is differences between tumors in rates of cell division and growth and in the capacity for metastasis and invasion of neighboring tissues. So we're going to talk about what are tumors, what are benign tumors, what are malignant tumors, what is a primary tumor, what is a secondary tumor, what is metastasis, and we need to distinguish between tumors that are and are not cancerous. And there is an application of skills which students should observe populations of cells to determine mitotic index. So through this video we're going to highlight cyclins, we're going to talk about checkpoints, we're going to talk about mutations and the consequences of those and how they regulate the cell cycle. We're going to talk about all things tumors and cancer. So we're going to start this video by talking about what cyclins are. And cyclins are a group of proteins that control the cell's progression through the cell cycle. Cyclins are proteins that are going to be substrates that a particular enzyme called a CDK, or a cyclin-dependent kinase, is going to act on. And so in the first phase of this reaction, the cyclin binds and activates the CDK. CDK is called cyclin-dependent kinase. Cyclin-dependent kinase, CDK, are the enzymes, whereas cyclins are the substrates. So the substrate is the cyclin, the enzyme is the CDK, and when the substrate and enzyme bind, it produces that enzyme substrate complex, and that enzyme substrate complex is a group of proteins that are going to be activated and induce some kind of metabolic reaction. The cyclin phosphorylates the CDK, enabling the CDK to act like an enzyme. What does phosphorylate means? It means the cyclin in this case is going to allow the CDK to bind to a phosphate. So it's going to add that phosphate to the CDK, and when it does that, the phosphorylation of the CDK is actually going to turn on or activate that particular enzyme, and it's going to allow it to behave like an enzyme, which, if you recall from our enzyme lecture, is going to catalyze a biological reaction. Remember, catalyze or catalyst means to reduce activation energy and speed up the reaction. Next, the CDK activates target protein to regulate the cell cycle, and so these activated enzymes then cause the cell to move through the cell cycle phases, G1 to S, G2 to M, or through the M phase, PMAT, prophase, metaphase, anaphase, telophase. So number one, phosphorylation of CDK and the cyclin binding to activate CDK. Two is CDK activates the target protein. The target protein is a protein that is going to help regulate the cell cycle. And then in step three, cyclin levels fluctuate throughout the cell cycle because the cyclin will be destroyed. The only thing the cyclin is produced to do within your body and within your cells, within your tissue cells, is to fluctuate in concentration. So as cyclin concentration goes up, its collisions naturally are going to increase, and it's going to increase the CDK conversion rate or the CDK activation rate. And once you use them, then you destroy them and you produce new ones. So That is the series of events or the progression that's going to help activate these target proteins, and it's these target proteins that are going to actually regulate the cell cycle. So when we talk about cyclins and their control over the cell cycle, remember the cell cycle consists of different phases. So we have G1, S, G2, and M. G1 stands for gap1, S stands for synthesis, G2 stands for gap2, and M stands for mitosis. All of those phases constitute the cell cycle. And then you'll also notice that there are a variety of cyclins, cyclin A, cyclin E, cyclin B, and cyclin D, that are going to fluctuate within this cell cycle. And so the concentration of these cyclins is going to increase and decrease or fluctuate over the course or the entirety of a cell's life cycle. So the points where CDKs function are called checkpoints. And you'll notice that there is a G1S checkpoint, which is the regulatory checkpoint that will allow a cell to progress through G1 and enter S. There is a G2M checkpoint, which again is a checkpoint that will allow a cell to progress from G2 into M phase. And then there is an M checkpoint, which will allow it to progress through mitosis, specifically from prophase, metaphase, anaphase, and telophase. All of these checkpoints are there to help regulate the cell cycle and to ensure that all of the cellular components are present and all of the processes are in place in order to allow the cell to progress to the next phase. Some cells will pause during this G1 phase and they'll enter a separate phase known as G0. And I know that we've talked about G0, but G0 is where a cell goes that reaches, or once it reaches, a state of stasis, which basically means if a cell is not going to actively divide or actively go through the entirety of the cell cycle, if it's a specific cell like a brain cell or a nerve cell, even if it's a cell that doesn't divide that often, like a muscle cell, it can reach this G0 phase, which is kind of like a roundabout. Some cells will enter this G0 phase and will eventually leave the G0 phase before it progresses through the cell cycle, but some cells actually go into this G0 phase and never come out, which is one of the reasons why brain injuries and spinal cord injuries are such bad injuries, is because a lot of times those cells can't replicate themselves and produce new ones. The cyclin levels change during the cell cycle. When a particular cyclin concentration is low, it binds to so few CDKs that the checkpoint cannot be reached. When you look at a graph like this, you need to think about these being thresholds, where if the concentration gets big enough, then it allows the checkpoint to be passed. If you think about these checkpoints like gates, then once these thresholds of cyclin and CDK levels reach a certain threshold, it opens that gate, so to speak, and allows the entire cell cycle process to progress through the gate. When its concentration reaches a certain threshold, the cell can move to the next phase, again, just like going through that gate. The G1 cyclin, so this is cyclin E, tells the cell to grow and to get ready to replicate the DNA. So the G1S checkpoint, remember what happens during synthesis phase, DNA replication. So if DNA replication occurs during S phase, it makes sense that the G1S checkpoint would have to make sure the cell has all of the enzymes necessary, the topoisomerase, the helicase, the RNA primases, the DNA polymerase 1 and 3, the ligase. All of those enzymes have to be present and have to be in proper concentration. G1S checkpoint requires the threshold of that cyclin E be such that it allows that gate to open and allows the cell to progress through that checkpoint in order to enter S and therefore replicate the DNA. G2 cyclin, which is cyclin A, ensures DNA replication occurred correctly, as well as to inhibit the DNA from re-replicating. Cyclin A also begins the transition from S phase to mitosis. Again, if DNA replication occurs during S phase, then that cyclin A, which you'll see, cyclin A, blue, is the next one. Cyclin A, once it reaches that threshold, will open the gate and will allow the cell to progress through S into G2 so that it is ready for mitosis. Mitotic cyclin, which is cyclin B, tells the cell to start making the microtubules that will form the spindle fibers for mitosis. Remember our discussion of mitosis. Spindle fibers are crucial not only in aligning chromosomes correctly, pulling sister chromatids apart, but also elongating the cell and constricting the cell in the middle, that contractile ring, that will allow the cleavage furrow to form, and eventually cytokinesis will take place. So there are several cyclins that have to reach certain thresholds in order to open gates that will allow the cell to progress through these checkpoints, which ensures not only the things that are supposed to be in place are in place, but allows the cell to progress knowing that everything that it needs for the next phase is present and is in proper concentrations. So what happens when you have cells that can and can't regulate themselves? There are two types of genes. We have proto-oncogenes and we have oncogenes. Proto-oncogenes are genes that can regulate the cell division. Notice these are genes that are going to be present in chromosomes that will result in normal cell division. Once those proto-oncogenes become mutated or overexpressed, they become oncogenes. Okay? A mutation exists in that proto-oncogene, and once that mutation exists, it will result in uncontrolled cell division, leading to tumor formation. Notice that when cells divide normally, they will produce normal, nice, neat tissue layers. However, if those tissue layers or cells become unregulated, they grow out of control and lead to masses that we call tumors. Now, tumors don't necessarily always mean cancer, but tumors mean mass or masses. So, proto-oncogene is a gene that can turn into an oncogene. Oncogenes contribute to normal cell becoming a cancer cell and can cause cells to divide more frequently than they should. Proto-oncogenes are normal regulators. Oncogenes are mutated regulators. Okay? Mutated genes turn into oncogenes. Typically, cells have their own death pre-programmed in their DNA so that when they no longer function, they can be broken down and their materials recycled in the organism. We call that apoptosis. Sometimes you hear that cell suicide or programmed cell death. However, oncogenes can modify apoptosis so that instead of dying and being broken down, the cell keeps dividing using mitosis. And if it keeps dividing when it shouldn't, that's how this mass of cells becomes produced. So this is a little diagram to show you kind of how a proto-oncogene transitions to oncogene. You have DNA. Part of the DNA is a gene. This particular gene in yellow is called a proto-oncogene. It is normal. It will result in normal cell division because it regulates cell division and regulates the cell cycle. However, if a mutation happens in that proto-oncogene, it turns the proto-oncogene into an oncogene, and that deregulates the cell cycle and produces a cell that can't control itself, which results in deregulated cell division, which can result in a mass known as a tumor. The oncogenes may start to change or mutate because they are triggered by an outside agent referred to as a mutagen. A mutation is a change in DNA sequence. A mutagen is an agent that results in mutations. So an example of a mutagen would be like cigarette smoke or UV radiation. Okay? Carcinogens, lead are all examples of mutagens that can cause mutation. So when we look at this particular diagram, you can see something called TSGs. TSGs are tumor suppressor genes. TSGs are genes that help prevent cancer by regulating cell growth and division by acting as brakes on cell growth. So you naturally possess TSGs, tumor suppressor genes. These are regulators that put the brake. So these are the brakes. TSGs control cell cycle progression, induce DNA repair mechanisms, promote apoptosis in damaged cells, and prevent uncontrolled cell proliferation. So again, these are the brakes. These are good. These help regulate the cell cycle naturally. Normal proto-oncogenes function by regulating cell growth and division, essentially acting as a gas pedal. So this one is a gas pedal. Notice the brakes are red and the gas pedals are green. Normal proto-oncogenes encode proteins that stimulate cell division, inhibit cell differentiation, and prevent cell death. So as a cell goes through its cell cycle, it's going to be pushing on the gas, which is proto-oncogenes. These are positive regulators. This is the gas pedal. It's green. They're going to naturally be progressing the cell through the cell cycle. You naturally also have these TSGs, tumor suppressor genes, which are acting as a brake, and so they are preventing a lot of the unwanted stuff from happening, while the normal proto-oncogenes are progressing naturally through the cell cycle. However, when the tumor suppressor genes become mutated, mutations in those tumor suppressor genes can disrupt their normal function, increasing the risk of cancer. Imagine you change the function of the brakes and they no longer brake your car or stop your car or train or bus or whatever effectively. You're not going to be able to brake or regulate through the cell cycle, and that becomes a problem. When proto-oncogenes undergoes mutation, again, now we're talking the mutation now exists in the proto-oncogene, which now we have a mutation in our gas pedal. We alter the gas pedal function. It can become an oncogene, leading to abnormal cell growth and cancer development. So in this analogy, we have a normal car that has normal brakes and normal gas pedal. We can get to where we need to go effectively because we have a properly functioning gas pedal and a properly functioning brake. When we become too fast or we are progressing through the cell cycle too fast, we have a brake pedal that will effectively slow us down and make sure we have everything in place in order to get to where we're going safely. Now let's disrupt this car and say now we're not going to have a functioning brake and we're going to have an accelerator or a gas pedal that sticks. If we speed up or we stick this gas pedal and we don't have brakes to slow us down, what's going to happen to our car? It's going to go out of control and it's going to be unstoppable. And so that's what cancer is. The cell cycle progresses too fast and we don't have a gas pedal to slow us down, which results in abnormal cell division, and it leads to a cancer. And it's one of the reasons why a tumor becomes such a big mass. It's because these cells are spiraling out of control. They are in a runaway car with no gas pedal, and it leads to unregulated cell growth or cell division. Some mutations in these tumor suppressor genes can happen during a person's lifetime, while others can be inherited. There are some cancers that are more inherited than others, and there are some that just happen during a person's lifetime as a result of mutagens or extended exposure to mutagens in their environment. So now we've talked about how cancer happens or how tumors happen, so let's talk about the differences between primary and secondary cancers as well as metastasis. A primary tumor is one that occurs at the original site of a cancer. So if this is where the gene mutates and therefore produces the first round of cancer cells, that is called primary cancer. A secondary tumor is a metastasis, a cancerous tumor that has spread from the original location to another part of the organism. Once you have a primary cancer and those cancers get from the primary spot into the bloodstream will transport the cancer cells just like it normally does. Like your bloodstream is a transport medium, blood is a transport medium, and your bloodstream can actually transport and place the tumor cells in a different location. That is a secondary tumor. That is what metastasis is. It's a spreading of cancer cells from a primary location to a secondary location. The difference between benign and malignant is that a tumor is said to be benign if the cells are forming a mass but are not spreading to the rest of the body. So if the cancer cells exist in a mass and it is not spreading, it's a benign tumor. If the cells are not contained within a mass, if they're growing in a mass, but they do have the ability or they have spread, it's said to be malignant. Malignant cancers can spread to the rest of the body, and malignant cancers are more difficult to treat and deal with than benign tumors are. And the last thing we need to do, this is the application of skill, is calculate a mitotic index. So this image shows you kind of an artistic rendition of the way a sample of cells would look under a microscope. This is a plant cell tissue. Notice the thick cell walls as well as the very uniform kind of block shapes that these cells are. You'll also notice that all of these cells have nuclei, and these nuclei are in different stages of the cell cycle. And so the first thing when calculating a mitotic index is we have to count the total number of cells, and we have to count the total number of cells that are in mitosis, and specifically in each phase of mitosis. So if we count the number of cells in mitosis, you can see the ones that are in mitosis are the ones in green. And I count this sample, this field of view, to have 20 mitotic cells. Okay, 20 cells in mitosis. I also count that there are 55 cells in interphase. And so if we add 55 to 20, that gives us 20 cells in mitosis over a total of 55 cells that we counted on this slide. If we divide 75 by 20, that gives us 0.2667, and if we round, that rounds to roughly 27%. 27% of these slides are in mitosis. Now, like I said at the beginning, we can even further drill down to the number of cells in each phase. And if you counted the number of cells in each phase, that would even give you a percentage of the time that the cell spends in that phase. And so basically what this number is telling us is that the cell spends roughly 27% of its life cycle in M phase. If we were to count maybe 5 of the cells out of 75, maybe it would give you a 5%. Maybe it would give you an 8%. And so you could say 8% of the cell's life is in prophase, for instance, if 5 of these cells are in fact in prophase. A mitotic index of 0.27 is low. It is unlikely that this tissue is undergoing rapid growth, so this tissue would be diagnosed as cancer-free. If it had a mitotic index higher than 75%, the doctor would be much more likely to consider the tissue as cancerous. Why do you think it's important as an oncologist or a doctor that specializes in cancer to look at tissue samples and compute a mitotic index? Normal tissue cells would have a very low mitotic index because if you think about the cell cycle, remember the cell cycle consists of basically 2 phases. You have interphase and you have M phase. If you were to look at this, about 25 to 27% of this cell by pie chart, by percent, would be M phase. So you would expect this mitotic index to be very indicative of a normal cell. If the cell is unregulated and is producing too much cell division, you would expect the cell to be in M phase longer, or more cells in M phase, because it's speeding through the interphase and replicating itself in M phase. So you would expect this mitotic index to go way up in as high as 75%. That's it for this particular video. We talked about a lot. We talked about cell cycle regulation, cancer, metastasis, and calculating a mitotic index. If you learned something, give it a thumbs up, subscribe to the channel, leave a question in the comments. See ya.